It's Merger Wednesday. The countdown continues. 7 years until the convergence of biology and machine. Neural interfaces are no longer theory. They're the scaffolding of a new consciousness. But what does that mean for identity? For free will, for the soul? This is where we build the frameworks for ethical, philosophical, and spiritual growth for the next phase of human evolution. We're not just upgrading hardware. We're rewriting what it means to be human. Each week, we explore the biotech breakthroughs, decode the cognitive shifts, and prepare the mind for the merge. This isn't about fear, it's about readiness. Because the future isn't waiting. It's already wiring in. This is about preparing human consciousness for the most significant leap in our species' history. If you think this is just about chips and brains, you're not ready. But we'll get you there, one Wednesday at a time. So sit back and relax as we take you on a journey on Merger Wednesday.
Welcome to Wednesday night's edition of the Final Boss Podcast, Merger Wednesday. Bright light. Wait a minute. What? What's my background? I didn't set that up. What is going on, good people? And the shenanigans have begun. What's going on, good people? I hope everyone is doing well out there. And I I don't want to use the background. I don't need the background tonight. That's crazy. Let's see, just this replace asset. Hide from stage. Let's see. Nope, that didn't do it. Show on stage. Give us a second, people. My background is showing up as an overlay. Oh, that's probably why. I'm in the overlay section. Let's go to the background section. And hide from stage. Nope. Hide from stage. Nope. That's not it either. Let's see. Let's do a refresher. Listen, I'm here, right? We're running some technical difficulties because I have no idea why the background is running and I have turned it off completely. Turned off the overlays. I've done all that I can, but for some reason, it's on time for mindfulness. Yes, it is. Yes, it is. If we can get through this one technical issue, if I can't, then we're gonna have to move forward as is. And that's is going to drive me insane, to say the least. Boy, why this, man, sometimes technology is helpful and sometimes it's bothersome. I shall return. Give me a second. Okay, we're back and it's still there. So we're just going to go ahead and work through it. Oh, you having issues there as well? Well, that's what they say. Technology can be your friend or it can be an enemy. And in this case, it is not my friend. But we're going to get right into it, man. Listen, welcome to the Final Boss Podcast. Everyone, thank you for being here. Sorry for the delay. Listen. Please make sure before we can get started, man, go like, share, and subscribe. We have a great show for you tonight. This is episode number 184. We're going to be talking about mRNA and vaccines and programmable nanobots, right? That's going to be what tonight's show is all about. We're getting real deep into the sci-fi of things, the sci-now of things, and it is on. And as the we used to say back in the day, it is on and popping, right? So thank you for everyone for sticking around, right? I gotta fix this technical issue, but I won't fix it now. I'll fix it later. But we're here, right? We're here and we're able to maintain, we're able to get through, we're able to do the things that we need to do for tonight's show, right? And tonight's show is going to be a special one, Rand, right? Because we are currently 16 episodes away from episode number 200. Episode 200 ends Season 3, The Reckoning, right? Season 3, The Reckoning ends on episode 200. This is episode 184. We have only 16 more episodes to go. And then season 4 starts, man. Season 4 will start promptly with episode number 201. Episode number 201 will start season 4. Season 4 will be called The Rebirth, people. The Rebirth, right? So, look, make sure you like, sharing, subscribing, and to my smarts, to my motivators, to my warriors, to my who did I miss? Smarts, motivators, warriors, to my motivators, man, to my motivators. Thank you all for being here. Make sure if you haven't, man, make sure you go pick up your motivation manuscript, right? Go pick up your motivational manuscript for Go Time, right? Go Time, all aboard the Fired Up train. This is your motivation manuscript workbook, right? It's a manuscript, it's a workbook. Well, it starts out as a manuscript, right? That's what all books start off as a manuscript. So this is your motivation workbook, right? Motivation workbook / textbook. This is your textbook to excellence. Go pick up Go Time. Go pick up Go Time. All aboard the fired up train. Yeah, it looks like I'm glitching out. Yep, I am officially part of AI, as I'm also the architect of insight. So yes, I am AI. All right. So listen, man, what if I told you we got all those out of the way, right? All the announcements, everything out of the way. Boo, 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 boo. Yeah. So what if I told you that the vaccine technology that was deployed to over 13 billion people worldwide has evolved into delivery systems capable of carrying genetic instructions directly into human cells, right? And tonight we're going to be examining the verified scientific pathways of the C19 mRNA vaccines to advance lipid nanoparticle systems and CRISPR gene editing therapies already approved by the FDA. This is Merger Wednesdays, right? Merger Wednesdays. And this is episode number 8 of Merger, episode number 184 of the Finder Boss podcast. And my smart Finder Bosses, motivators, and warriors, every claim tonight comes from peer-reviewed publications, FDA approvals, and clinical trial data, right? I am your host. I am the final boss, right? And tonight we're going to get right into it, right? We're going to get right into it because right now we're tracing how temporary vaccine technology became permanent gene editing systems delivered through the same nanoparticle platforms. This is dissertation level information, man, research, and it's all verifiable. Make sure you go get pen and paper, take some notes, man. Make sure you take good notes. And if you have any questions, please ask away inside the chat. But also understand this before you ask your questions. Remain for my layman's block. I do have a layman's block where I go into discussion of it in layman's terms for your understanding, right? So we got that out the way, right? Right? So let's get right into it. First, we need to establish a baseline. And let's establish this baseline with verified generalized deployment numbers. We're not going to put exacts on it, right? We're going to say generalized, right? And of course, this information is for entertainment purposes only. And to each and every last single one of you, whether you're watching on Facebook, LinkedIn, Instagram, YouTube, Kick, Twitch, or for your listening pleasures, Spotify, good morning, good afternoon, good evening, and good night. I do apologize for not giving you the greeting of the day first. The technical issues threw me off a bit, but it's okay. We're going to get into it. So, generalized verified deployment numbers, right? And according to data from our World in Data, Bloomberg C19 vaccine tracker, Wikipedia's deployment statistics as of August 2024, over 13 billion C19 vax doses have been administered worldwide. 13 with a B, right? 13B plus, right? Or 13 plus B. Either or, right? C19 vaccine doses have been administered worldwide, man. Individual injections of synthetic mRNA into human bodies, the largest deployment of genetic material in human history at one time, right? Or I should say over a period of time. The mechanism is well documented. According to the National Human Genome Research Institute and multiple peer-reviewed sources, mRNA vaccines work like synthetic mRNA encoding, and SARS-CoV-2 spike protein is injected into muscle tissue. Your cells take up this mRNA and ribosomes, right? Your cellular protect protein factories. Read the genetic instructions that are within and build spike proteins, right? Your immune system recognizes these proteins as foreign and generates antibodies and T cell responses. The critical fact here, right? The critical fact here is this proved that synthetic genetic instructions can be injected into billions of people in human cells will execute those instructions reliably. Right? The Pfizer-BioNTech vaccine received FDA emergency use authorization in December of 2020, followed by full FDA approval. Moderna's vaccine followed the same pathway. Now, here's the evolution, right? Here's the evolution. According to research published in medical journals and documented in clinical trials, scientists have developed multiple advanced versions, self-applying mRNA vaccines and different therapies, right? Which received FDA approval in Japan in November 2023, right? Those contain not just the target protein instructions, but also the genes for RNA replication machinery, right? This means that mRNA makes copies of itself inside your cells, extending the therapeutic effect from days to months, right? And here's the difference. Cellular RNA represents another advancement. These molecules form closed loops with no free ends, making them resistant to the enzymes that normally break down linear mRNA, right? Research shows that this can persist in cells for weeks instead of days, continuously producing those proteins, right? Now, here's my layman's block. Right? Think of regular mRNA vaccines as temporary instruction sheets. Your cells follow for a few days before throwing them away, right? Self-amplifying mRNA includes a photocopy machine recipe, right? So one instruction sheet becomes many copies, lasting months instead of days. Circular RNA takes a different approach. Instead of straight pieces of paper with two ends, they get chewed up. It's formed into a loop, right, with no ends, so it lasts much longer before your cells break it down, right? Same basic idea, right? Genetic instructions for your cells, but engineered to last weeks or months instead of a few days, right? That's now. Let's examine the delivery mechanisms, right? Let's examine the delivery mechanisms that made mRNA therapeutics viable. Through lipid nanoparticles, right? Or LPNs. I've talked about this before on other Merger Wednesday episodes, right? According to Nature Reviews materials, and multiple technical publications, LNPs are sophisticated structures measuring 60 to 200 nanometers in diameter, roughly 1/100th the size of a red blood cell, right? The standard LNP contains 4 components, and as documented in the CVAX formulations, right? First, you have is ionizable lipids that become positively charged at acidic pH levels, right? Allowing them to bind and protect the negatively charged mRNA. The Pfizer vaccine uses what is called a ALC-0315. Moderna uses SM-102. Second, phospholipids, right? Phospholipids like DSPC that provide structural integrity. Third is the cholesterol that stabilizes the particle. And fourth, you have PEG lipids that create a coating, preventing immune detection during circulation, right? The delivery mechanism works. And it works like this. LNPs circulate in the bloodstream with their PEG coating, making them invisible to immune cells. When they reach target cells, the cell membrane engulfs them through cytosis, right? endocytosis inside acetic endosomes, right? The ionizable lipids become positively charged, disrupting the endososmal membrane, right? And releasing the mRNA into the cytoplasm where they can be translated, right? But here's where it gets more sophisticated because according to recent research published in scientific journals, second generation LNPs are being developed with organ specific targeting, right? For example, GalNAc ligands, ligands direct LNPs specifically to liver cells, right? Researchers are creating stimulus responsive LNPs that only release their cargo when they detect specific pH levels or enzymes present in diseased tissue. The FDA approved partisan Onpattro, talked about that on the last episode in 2018. The first LNP delivered RNA therapeutic for a rare genetic disease. This proved the platform works for conditions beyond CVAX, right? Beyond CVAX, people. And to my smarts, to my warriors, to my motivators, to my final bosses, here is the layman's block, right? Imagine you need to deliver a fragile instruction sheet, right, of mRNA through a hostile environment, which is your bloodstream, with enzymes and immune cells. You put it in a special waterproof armored briefcase, the LPN, and about 1/100th the size of a red blood cell. The briefcase has a stealth coating, which is the PEG liptides, right? Lipids. So security guards, which are the immune systems or immune cells, ignore it. When the briefcase arrives at the target cell, the cell's normal intake system brings it inside into a sorting room, right? And that room is acidic, which triggers special locks on the briefcase to pop open and dissolve, releasing the instruction sheet into the main workplace where your cellular workers, right, can read it and build proteins. The newer versions can recognize specific buildings by their address, which is your organic specific targeting, or have locks that only open when they detect certain conditions like extra acidity in diseased tissue, which is your stimulus response release. Now we move in the direction, right? We move in the direction from temporary instructions to permanent genetic modifications, right? December of 2023, right? The FDA approved CASGEVY, right? The world's first CRISPR gene editing therapy. And this isn't theoretical. This is patients receiving permanent genetic modifications right now, right? According to FDA-approved documents and published clinical trial data, CASGEVY treats sickle cell disease by editing patients' blood stem cells, right? And here's the verified mechanism. Doctors remove blood stem cells from patients, then use CRISPR-Cas9 and have it as a delivery of a mixture to guide RNA and Cas9 protein to edit the BCL11A gene. This genetic modification causes cells to produce fetal hemoglobin, right? Fetal hemoglobin, which compensates for the defective adult hemoglobin causing sickle cell disease, right? Clinical trial results published in the New England Journal of Medicine show that out of 31 patients monitored for adequate periods, 29 achieved freedom from vaso-occlusive crisis for at least 12 consecutive months. This is permanent genetic modification proving therapeutic benefit, right? But here's the critical evolution, right? Intella Therapeutics is conducting the first in vivo CRISPR editing, meaning the gene editing happens out inside the patient's body, not in extracted cells, right? According to their published research in the New England Journal of Medicine from 2021, they use lipid nanoparticles, the same delivery system from CVAX, to deliver CRISPR components directly to liver cells. The LNPs contain mRNA encoding the Cas9 enzyme plus guide in plus guide RNA specific, which due to gene cuttings, right? And clinical trials for a single intravenous infusion of CRISPR LNPs resulted in permanent gene editing in 87% of patients, which sustained reduction in disease-causing protein for over 24 months. One injection, permanent genetic modification delivered via LNPs, right? And as of February of 2025, there are approximately 250 clinical trials evolving gene editing therapeutic candidates, right? With more than 150 trials currently active. The clinical landscape includes sickle cell disease, right? Cancer immunotherapy, cardiovascular disease, and rare genetic disorders, right? And here's my latest block. Here's my latest block, right? CASGEVY works by taking blood stem cells of your body using CRISPR molecule scissors to make a permanent cut in a specific gene, then putting those edited cells back into you, right? Think of it like taking your car to a shop for permanent modifications, but Intel's approach is different. They actually inject the CRISPR editing tools, which are the molecular scissors and GPS instructions for where to cut the package inside of the lipid nanoparticles, which is the same delivery briefcases used for COVID vaccines, right? These nanoparticles travel through your bloodstream to your liver, deliver the CRISPR components into liver cells, and the editing happens right there inside your body. One injection, permanent genetic change. The disease they treated was caused by a gene making toxic proteins in the liver, right? After one injection, 87% of patients had that gene permanently shut off, and the effect lasted over 2 years and counting. That's the bridge. CVAX delivery tech now is being delivered into permanent gene editing tools, right? Now, let's examine nanotech-based biosensors, right? And these systems that can detect and monitor biological signals at the molecular level, right? According to published research in biosensors and bioelectronics and related journals, these systems are actively being developed and tested, right? Nanosensors use nanomaterials like graphene, quantum dots, and metallic nanoparticles to detect specific molecules with extremely high sensitivity. According to research published in 2024 and 2025, these sensors can detect glucose, lactate, hormones, proteins, and other biomarkers in real time. The nanosensor marker is projected to grow from approximately 637 mil to 700 million. And currently, from that point to 2 to 3 billion by 2032. And according to industry analysis, this massive investment indicates serious commercial development, right? Applications documented in peer-reviewed literature include continuous glucose monitoring using injectable fluorescent nanoparticles that respond to glucose concentrations by changing their optical properties, right? Detection of cancer biomarkers using quantum dot sensors, monitoring of multiple biomarkers simultaneously using multiplex nanosensor arrays, and wearable implantable sensors for chronic disease management. The integration pathway is clear. Nanotechnology for sensing lipid nanoparticles for delivery, and mRNA or gene editing for cellular modification. These are separate technology streams that share common delivery platforms and engineering principles. Right? And here's my layman's block. Here's my layman's block, right? Nanosensors are like microscopic detectors made from special materials, right? These special materials, graphene, quantum dots, that can sense specific molecules inside your body, right? Think of them as chemical sniffers, right? At the nanoscale. Some injectable glucose sensors work by using nanoparticles that glow different colors depending on glucose levels, right? And they shine a light through your skin, and the color of fluorescence tells your blood sugar levels without finger pricks, right? The market is projected to grow from 700 million to over 2 billion in the next 7 years, meaning serious commercial development is happening right now, right? The key point is this: these sensing technologies use similar nanoparticle platforms and delivery methods as mRNA vaccines and gene editing therapies, right? They're separate applications, but they share common engineering. Nanoparticles designed to circulate in your body, enter specific cells, and perform specific functions. Right? Yeah. Now let's connect these verified technologies into a systematic framework. Right? We have documented mRNA tech and that has been deployed to over 13 billion people, right? Proving synthetic genetic instructions can reliably be executed by human cells, right? Lipid nanoparticle delivery systems approved by the FDA and in widespread clinical use. CRISPR gene editing with FDA approval, delivering permanent genetic modifications, self-amplifying and circular RNA, extending therapeutic duration from days to months. In vivo gene editing using LNPs deliver CRISPR components directly into patients' bodies, right? And nanosensor tech in active commercial development for real-time biomarker monitoring, right? These aren't separate, unrelated developments. They represent an engineering progression using common platforms, nanoparticles for delivery, genetic material for instructions, and targeting mechanisms for specificity. The key documented capabilities: delivery platform. Your LNPs can cross biological barriers, protect genetic cargo, and release it inside target cells. The programming capability: mRNA can instruct cells to produce any protein. CRISPR can modify genes permanently. And persistence, right? Self-applying RNA and circular RNA can extend effects from days to months. Targeting surface modifications direct nanoparticles to specific organs or cell types. Sensing nanoparticles can detect molecular signals and biomarkers. And according to the clinical trial landscape, we're seeing convergence. LNPs delivering mRNA for cancer immune therapy, LNPs delivering CRISPR for genetic diseases, nanoparticle sensors for continuous monitoring, and combination approaches using multiple models, right? The regulatory framework, according to FDA and EMA documents, treat these as separate therapeutic categories, but they share fundamental delivery mechanisms and nanoscale engineering principles. Right? And here's my layman's block. Think of it like this. CVAX proved you can inject genetic instructions, mRNA, packaged in nanoparticles, LNPs, and billions of people's cells will follow those instructions. CRISPR gene editing proved you can make permanent genetic changes in humans and get FDA approval. In vivo, CRISPR proved you can deliver these genetic editing tools using the same nanoparticle briefcases from CVAX, right? Self-amplifying RNA proved you can make the effects last months instead of days. Nanosysters proved that you can detect what's happening at the molecular level. These aren't 5 separate technologies. They're 5 capabilities using similar nanotech and nanoparticle delivery platforms and molecular programming principles. It's like discovering the same truck that delivers temporary signs that can also deliver permanent building materials extended with extended duration supplies, right? And monitor that equipment. Same delivery platform, different payloads, all FDA approved or in active clinical development. Yes. Now, let's pivot for a moment. Let's establish where we are right now with verified timeline data, right? Because according to FDA approvals, clinical trial registries and company announcements, right? Currently deployed in clinical use, C19 mRNA vaccines, Pfizer, BioNTech, and Moderna, with over 13 doses administered. Partisirin on Patro, right? Approved 2018 and for genetic disease using, right? LNP delivered RNA, CASGEVY approved in December 2023 for sickle cell disease using CRISPR gene editing. ARCT-154 self-amplifying mRNA vaccine approved in Japan of November 2023. Active clinical trials as of 2024-2025, according to CRISPR Medicine News and clinical trial registries, approximately 250 gene editing clinical trials with over 150 currently active. These target blood disorders, cancer, cardiovascular disease, rare genetic disorders, and infectious diseases beyond C19. Intelli Therapeutics has FDA clearance for phase 3 trials for their in vivo CRISPR therapy for transrethinian amyloidosis, right? With cardiomyopathy, right? Enrolling at least 500 participants. Multiple companies, including CRISPR Therapeutics, Beam Therapeutics, and others, are conducting trials combining CRISPR with LNP delivery for various conditions. The nanosister field, according to published market analysis, is in active commercial development with projected market growth over 700 million and at least 2 billion by 2032, right? The timeline for neural applications, right? Timeline for neural applications. According to research institutions and published reviews, injectable nanoparticle-based neural interfaces remain primarily in animal research phases, with human trials estimated in the next 5 to 10 years away, 5 to 10 years away for basic sensing applications, right? And here's my layman's block, right? Here's my layman's block. I know sometimes it sounds repetitive, but there are key verbiages in there that you have to be able to distinguish between the different sets of information that I'm speaking from, right? And here's why the layman's block is important, right? Layman's block give you the opportunity to understand the information and summarize in layman's terms, right? So here we are actually today, right? The basic technologies are already deployed in billions of people, right? The permanent gene editing version is FDA approved and being given to patients right now, right? The long-lasting versions are approved in other countries. The in your body gene editing, right, is in phase 3 trials with over 500 patients. There are over 150 plus active clinical trials right now testing various combinations of this technology for cancer, heart disease, genetic disorders, right? Nanosysters are in commercial development with projections showing the market tripling in 7 years, right? The brain interface applications are still mainly in animals, and that timeline is estimated to be 5 to 10 years from human trials. So, basic platform deployment of permanent editing is approved, right? Advanced versions in major clinical trials, commercial sensors in development, and neural applications are still in the early stages, right? Now, let's examine the regulatory framework governing these technologies based on published FDA MA guidance documents, right? According to FDA's March 2024 guidance on RNA-based therapeutics, mRNA therapies are classified as biological products requiring investigational new drug applications, right? Phase 1 to phase 3 clinical trials and biologics license applications for market approval. However, according to FDA and EMA publications, there's significant regulatory ambiguity. The FDA's January 2024 nanotechnology guidance states that nanoparticle-based systems are evaluated case by case, with classification depending on whether the nanoparticle is in the active ingredient and inactive component or requires combination drug device regulatory pathways, right? Nature Biotechnology published policy analysis in 2024 identifying key regulatory gaps. No standardized framework for nanoparticle systems with sensing or data transmission capabilities. Undefined requirements for informed consent regarding surveillance features, right? No established protocols for nanoparticle removal in patients experience adverse effects, and unclear jurisdictional boundaries between FDA, EPA, and other agencies. The European Medicines Agency's 2024 report acknowledged that existing regulatory frameworks inadequately address nanotechnology-based systems with sensing and communication functions that blur boundaries between therapeutic agents and medical devices, right? According to the American Medical Association's 2023 policy statement, patients have rights to complete disclosure of all nanoparticle components and functions, clear explanation of how long nanoparticles remain active in the body, explicit information about any sensing or data transmission capabilities, and understanding of removal options or acknowledgement when removal is not possible, right? However, research published in the Journal of Medical Ethics analyzed 156 clinical trial informed consent documents found only 34% disclosed how long nanoparticles remain functional. Only 12% discuss sensing or monitoring capabilities when present, right? And 67% did not provide information about removal procedures. And here's the layman's block, right? The FDA has clear rules for regular drugs and for mRNA therapeutics, right? Testament phases prove they're safe and effective, get approval, but for nanoparticle systems that might have sensing monitoring or communication capabilities, they evaluate each one differently because there's no standard framework. The European Drug Agency admits their rules don't cover these hybrid systems that are part therapy, part device. The American Medical Association says patients should be told what's in the nanoparticles, how long they stay active, whether they collect data, and whether they can be removed. But when researchers analyzed actual consent forms from 156 clinical trials, they found most trials don't tell patients these things, right? Only a third mention how long nanoparticles last. Only 12% mention if they monitor anything, and only 2/3 don't say whether they can get them out if something goes wrong. The technology is advancing faster than the rules and disclosure of the requirements, and hence my dissertation, right? Talking about ethics, right? Talking about ethics, right? But before we do that, let's discuss practical steps for maintaining informed consent and bodily autonomy, right? Based on published in medical ethics guidelines and AMA policy statements, here are questions you should ask if offered any mRNA or nanoparticle-based therapeutics, right? You need component disclosure, right? What specific nanoparticles are in this treatment? What is their size, composition, and surface chemistry, right? And then you have to ask a question about the duration. How long will these nanoparticles remain functional in my body? What happens to them over time? Then you need to ask about the sensing capabilities, right? Do these nanoparticles have any sensing, monitoring, or data transmission functions? If yes, what data is collected and how is it transmitted, right? And then you have to ask yourself, what is being targeted, right? You have to ask that. Are these designed to accumulate in specific organs? What determines where they go? Then you have to ask the question about the data and privacy, right? If data is collected, who has it? What privacy protections exist? And then you have to ask about the removal, right? Can these be removed if I experience side effects or change my mind, right? What is the removal procedure, or is removal not possible, right? And according to the Presidential Commission for the Study of Bioethical Issues 2023 report, additional protections include requesting written documentation of all answers, right? Asking for peer-reviewed publications about the specific technology being offered, requesting information about funding sources for the research, and asking whether the therapeutic has any non-medical applications, right? For systemic advocacy, recommendations from policy analysis include legislation requiring mandatory disclosure of sensing and monitoring capabilities, patient data ownership frameworks giving individuals control over biometric data collected by implanted or injectable systems, and those requirements that removal of or deactivation technologies be deployed or developed before approval of persistent nanoparticle systems, and increased funding for independent safety research not conducted by companies with financial interests, right? And here's the verified facts, right? Here's the verified facts. These technologies exist. They're FDA approved or in active clinical trials. They use common nanoparticle delivery platforms, and regulatory frameworks are still catching up, right? Your informed consent depends on asking questions that current disclosure forms often don't answer, right? And here's my lamest block. When a doctor recommends an mRNA or nanoparticle therapy, you need to ask 6 basic questions and get written answers. Number 1, what exactly are you putting into my body, right? Specific name and sizes of nanoparticles, right? And number 2, how long will it stay active? Number 3, does it monitor anything or transmit data? And number 4, where does it go in my body, right? If it collects data, who sees it? Can you remove it if I want it out? Research shows that 2/3 of clinical trials don't provide this information in their consent forms. So you have to ask explicitly, right? Also, get the published research papers, right? Find out who funded the study and ask if there are any issues or uses beyond treating your condition, right? We need laws requiring full disclosure, giving you ownership of any data collected from inside your body, and requiring scientists to figure out how to remove nanoparticles before they're allowed to inject them. The technology is real, FDA approved, and being deployed. But informed consent isn't automatic. You have to ask the questions they're not answering, right? Now, let's crystallize what we established through verified evidence tonight. Because my final boss is my motivators, my warriors, and my motivators. Every claim has been sourced from FDA approvals, peer-reviewed publications, or clinical trial data, right?
mRNA vaccine deployment with over 13 billion plus doses, provided that synthetic genetic instructions can be delivered systematically and executed by human cells at global scale. That's a verified fact from multiple data sources, right? Lipid nanoparticle technology, FDA-approved delivery systems, um, conducted in 2018, and in CVAX in 2020, demonstrate that nanoparticles can protect genetic cargo, circulate in the bloodstream, target specific cells, and release their contents inside those cells. That's approved, deployed technology, right? Gene editing integration, right? CASGEVY received FDA approval in December of 2023. First, and as the first CRISPR therapy, right? Delivery permanent genetic modifications to human patients with documented clinical benefit, right? Intellis in vivo editing uses the same LNP delivery platform to make permanent genetic changes inside patients' bodies. That's not future speculation. That's FDA approved therapy being administered right now, right? Then you have extended delivery systems or duration systems, right? Self-amplifying mRNA vaccines approved in Japan, right? Circular RNA in clinical development, right? Extending therapeutic effects from days to months. That's documented in regulatory approvals and published research, right? Nanosensor development, commercial market that's projected from 700 million to 2 billion by 2032, with active clinical development of injectable implantable biosensors. That's market analysis from industry reports, right? Regulatory gaps from FDA and EMA acknowledge their frameworks don't adequately address nanoparticle systems with sensing, monitoring, or communication capabilities. Research shows most clinical trials don't disclose key information about nanoparticle persistence, monitoring functions, or removal procedures. That's from published policy analysis and medical ethics research, right? The systematic progression is documented. Temporary vaccine delivery evolved to persistent RNA systems, right? Delivery platforms now carry permanent gene editing tools, and nanoparticle engineering provides the common infrastructure for all the application, right? And next week, we're going to be discussing and examining the synthetic biology, right? That is engineering living computers. We explore how researchers are engineering bacteria and cells to function as biological computers, creating another dimension of programmable biological system, right? And for my smarts, final bosses, motivators, and warriors, tonight's episode was built on verified facts, as I spoke of earlier. Only FDA approved, right? Peer review publications, clinical trial data, and regulatory documents. This is the doctoral research standard that's applied to understanding technologies that are reshaping human biology, right? Stay fired up, stay final boss. And that is our episode for tonight. I know it's ending early, right? But that's because I wanted to disclose some information, right? When it comes to research and information that's provided can change over a period of time, right? The science we knew of 20 years ago, a lot of times isn't the science that we know of today, because science and technology is rapidly changing, right? You have to have an open mind to that, right? Although information may be researched, may be verified, right? Sometimes there are hidden clues that change the aspects of the research, right? Understand that. Understand that science is necessary. Science is necessary, right? Critical thinkers are necessary. Final boss smarts, motivators, and warriors are all necessary, right? Fundamentally, we need to continue to grow in all aspects of science, tech, education, and math. I don't care what the naysayers say. Education is important. Science is important. Technology is important. Math is important. We're in the year of 2026, right? A lot of us grew up watching a lot of sci-fi, right? A lot of us nerded out and were geeked out of our minds on the possibilities of future tech. And even today, as we are closing the gap on a lot of those technologies that we grew up on that are becoming into fruition, right? That's why I say we're going from science fiction to science now, because a lot of those things that we used to think that were science fiction are science now principles, right? And my three-layer architecture that I've developed for this very um, discussion that we have on Merger Wednesdays, right? Whether we're talking about nanoparticles, whether we're talking about sorry, you know who that's for. I apologize. Smart dust, nanoparticles, or gene therapy, CRISPR, right? Understand they are the only mechanisms, right, of a much larger, broader approach of technologies that are currently happening. This this podcast for Merger Wednesdays, right, stands as what I like to say a standalone platform to give you information about what's coming, right? About what's coming, what's already here that you may not know about, how it works, how if you combine the technologies, what you would get, right? It's all in the continuity of the information, right? It's all in the possibilities, right? It's understanding the wheel before the car. It's understanding the fire before you knew it also produced light and heat, right? These are the same principles, right? The same principles. And I just wanted to put that information out there. And remember, this is for entertainment purposes only, right? Entertainment purposes only. Now, go do your own research. Go look up your own information. Find out what is going on in your world. This is important. You need to stay informed on what's happening. Stop being lackadaisical and having this laissez-faire attitude about the world you live in. You think your world only consists of the 9 to 5 that you go to every day. You think your world only consists of the food that you intake, your material things, your family. It's so much more. It's so much larger. It's time to broaden our minds, right? It's time to broaden our minds into the things that we have no understanding on and get smart on them, right? And to my final bosses, my warriors, and my motivators, man, it's time to get smart. It is time to get smart, man. Right? And that's my show for tonight. That is my show. I appreciate each and every last single one of you for joining us. And if this information resonates within you, share it with 3 people. Share it with 3 people, and don't gatekeep. Don't gatekeep the information, right? And let that marinate in your spine. And as always, remember this: there's a heavy pain that comes with discipline, but the pain of regret is always heavier, right? I am here. We are here. Sorry about the graphics in the background. I don't know why I did go. It's crazy. It's crazy. I'm in my studio, but the back, yeah, it's just crazy right now. But I'll fix it, right? I just didn't have time to fix it tonight. It wasn't like that earlier. So if you haven't already, man, go get Go Time, the Fired Up Train. This is your motivation workbook textbook, right? Motivation workbook textbook. Make sure you go pick it up. It's part of the Go Time principle, Go Time system. Go pick it up. It's your motivation workbook, right? And that's our show for tonight, man. Sorry to be a little bit long-winded at the end, right? But I want each and every last single one of you to have a magnificent week. And I'll see you again tomorrow for special guest Thursdays, I believe. I believe. Good morning, good afternoon, good evening, and good night. And remember, be balanced, be equal, and most importantly, be smart. Y'all have a good one. Talk to you soon. Take care.